Good afternoon. In our last lecture, what we focused on was DNA structure, the chemical and physical properties of DNA as an independent entity. So today what we're going to do is focus on what I refer to as the structural organization of the genome. That is, how is the genetic material packaged or organized into chromosomes to fit inside the cell. But before we do that, I want to review just a little bit of the basics, the differences between prokaryotic cells and eukaryotic cells. So taking one step back, again, one of the comments or points that I always discuss in my introductory biology courses is that all cells, no matter if they are prokaryotic or eukaryotic, contain DNA at some point in their life cycle. All cells possess ribosomes, which are the structures that synthesize proteins, and all cells are bound by a membrane. So those are three characteristics of every single type of cell, again, whether it's prokaryotic or eukaryotic. But if we focus in or hone in a little bit more on the exact definition as far as prokaryotes and eukaryotes are concerned, the exact definition of prokaryote refers to not having a nucleus, whereas eukaryotes refer to having a true nucleus. So again, just some basic review. The nucleus is a membrane-bound organelle that houses DNA. It is in fact a double membrane system that houses the nuclear material, made up of a nuclear lamina, nuclear pores, and a nucleolus where ribosomes are synthesized. So this structure that specifically houses the DNA is something that is specific to eukaryotic cells. Prokaryotic cells, like I mentioned previously, also have DNA, but their DNA is not housed or packaged in a specialized structure to hold the genetic material. It is found in what's referred to as the nucleoid, which is just a region of the cell where the DNA or the chromosome can be found. So now that we've established some basic differences between prokaryotes and eukaryotes, let's still generalize and talk about the functionality of packaging DNA into a cell. So in eukaryotes, for example, this packaging is really important because you need to fit all of the DNA into that specialized structure referred to as the nucleus. In a typical human cell, we're talking about 2 meters of DNA that need to be coiled up and packaged inside this structure. In addition, packaging also provides protection of the DNA. This is really important because DNA is in fact the genetic material. It is the molecule that passes on information from generation to generation. And that DNA does in fact need to be protected in order to maintain the integrity of the genetic material from generation to generation. Lastly, packaging of the genetic material allows for organization. Think about simply taking your clothes, if you are trying to pack it into a suitcase, and just stuffing it all into a suitcase. Well, there's not a lot of organization there. If you're looking for something specific, you're going to have to pull everything out in order to find the one thing that you're looking for. So, in order to have some organization associated with this packaging, this is accomplished by a set of proteins that we will look at in both prokaryotes and eukaryotes in order to, again, completely organize that genetic information. One additional common feature that I want to highlight before we move on to looking at prokaryotes and eukaryotes specifically is that DNA is found in association with proteins. And when the DNA is found in association with proteins, this is now referred to as chromatin. It is the chromatin that condenses and allows for the material to be packed inside of the cell, whether it's prokaryotes or inside the nucleus in a eukaryotic cell, and allows for that protection and that organization that I was just referring to. So again, DNA is not typically found in just the naked, plain, double helical state. It is found in complex association with proteins, and at that point it's referred to as chromatin. So let's review really briefly one of the things that I mentioned in the last lecture, and that was prokaryotic genomes are typically found in a circular organization, which is distinct from most eukaryotic genomes, which are found in a linear organization. So what you see in this particular figure is a circularized piece of DNA that represents a prokaryotic chromosome. However, this is not a very efficient way of packaging the genetic material inside the prokaryotic cell. Even though there's not a specialized structure like the nucleus to house the genetic material, there does still need to be some compaction and organization to that genome rather than being in that circularized form. So what we oftentimes see with prokaryotic chromosomes is, yes, they are circularized, however, they are found in what's referred to as a supercoiled form. What typically happens is the circularized piece of DNA has a nick or a cut that's made in one of the strands, usually just on the sugar phosphate backbone, and that allows for relaxation or what's referred to as supercoiling of that circularized structure. And in the far right, you can see the result of that supercoiling of the genetic material. The best analogy that I usually like to use is to take a rubber band. If you take a large rubber band and hold it on either end of the circle and begin twisting one end, what you would begin to visualize is that that rubber band begins to coil and then it'll tighten up and sort of shrink in your hand. Hopefully you can all visualize that. One of the pitfalls of not being able to see you and demonstrate these types of things. But hopefully you can visualize that. And the idea there is that is what's happening when a circularized piece of DNA is supercoiled. One end is being held, the other end is twisted, and this then shrinks the DNA into the supercoiled form. Now, before I continue, one of the things I typically like to mention is that throughout the semester, what I oftentimes like to do is focus on the prokaryotic model for whatever topic it is that we happen to be discussing, and then add in a little bit of the eukaryotic information. The reason for this is that prokaryotes have been studied extensively, and we have a vast amount of knowledge and understanding of different processes in prokaryotes. Eukaryotes are much more complex, generally speaking, and typically include a lot of additional regulatory steps, which means a lot of additional proteins in the process of any particular function we happen to be talking about or mechanism we happen to be talking about. So again, for the most part throughout the semester, we will focus on understanding the prokaryotic model with a little bit of eukaryotic thrown in there. But for today's lecture, we're going to actually reverse that. And that's because with prokaryotes, there's not a whole lot in comparison to eukaryotes that's happening with respect to the packaging of DNA in the prokaryotic cell. So let's go ahead and start with prokaryotes then. Prokaryotes, again, do not have a nucleus. They don't have a specialized structure that houses the genetic material. They do have what's referred to as the nucleoid region, and this is where the DNA is found inside of the cell. However, there is still a lot of compaction that has to happen with that DNA in order to get it to fit inside the cell itself. The DNA must compact from that circularized structure that we talked about as being the basic structure for a chromosome. It must compact greater than 1,000 times to be able to fit inside of the cell. So how is that accomplished? Well, there's a number of components that we'll talk about with this particular slide that play a role in that packaging of DNA in prokaryotes. The first thing I want to mention is what I mentioned on the previous slide, is that the DNA itself is compressed due to the supercoiling that we were just talking about. So again, the rubber band analogy, you have a circle representing the rubber band, and if you twist that, it begins to sort of shrink down and condense into what would look like a supercoiled piece of DNA. So that is the first level of compaction that actually occurs in the prokaryotic cell. It is the supercoiling. Secondly, there are proteins that are playing a role in helping with this process overall. There is one protein that is referred to as HU, and HU is a protein that is involved in allowing for that compaction to take place. So let's look at these figures on this particular slide. The one on the far left is an actual image of a prokaryotic chromosome in its complex nature. And what it kind of looks like is just a bunch of squiggles that are all scrunched together right in the center. And hopefully I'm describing that well enough for you to see that in the left-hand image. What's allowing for that scrunching together to take place is depicted in the cartoon version on the far right. On the far right, you can see supercoiled regions or stretches of the DNA. Those are those supercoiled squiggles that you see. And then what's highlighted here on this figure just as proteins are those little yellowish cream color proteins sort of right at the center. Kind of looks like a flower structure right at the center of the flower. Those are the HU proteins. And those HU proteins are now bringing in separate components or separate arms, if you will, of supercoiled DNA to allow for that extreme amount of compaction that needs to take place. Now, what allows for this supercoiling is, like I mentioned on the last slide, is that a nick is made in the sugar phosphate backbone, and this is an enzyme known as topoisomerase. We will come back to topoisomerase many times throughout the semester, but this is an enzyme that does exactly that. It makes a cut in the sugar phosphate backbone, which can do lots of different things, but in this particular case allows for the supercoiling, the winding, and the unwinding to occur. And those HU proteins are what are sort of gathering those different arms together at the center. Now, there are some additional proteins that are not shown on this particular slide, but I do want to highlight them because they are important in packaging DNA in prokaryotic cells. In addition to HU and topoisomerase, which I just mentioned, there is a protein known as IHF, which is short for integration host factor. This is a protein that binds to specific sequences within the genome and allows for the introduction of additional bends in the structure. That's what you're seeing right in the center figure, is that complex of proteins that is allowing for the bending of the DNA overall. There are also a group of proteins known as the condensin protein complex. Condensin protein complex consists of FIS, which plays a role in activating transcription, and HNS, which also plays a role in regulating transcription overall. All right, so now let's transition to looking at eukaryotes in a lot more detail. In this particular figure, what we're doing is looking at an overview of that structural organization that occurs. It's a highlight of the things that are coming in the next several slides. So the first thing up at the very top is the double helix DNA, and this is what I sort of jokingly refer to as naked DNA, just the double helix. We know that DNA is not found in this naked state. It is found in complex association with proteins in eukaryotic chromosome structure. Again, the DNA and the proteins together are referred to as chromatin. That DNA association with proteins is that first level of organization that you see that's labeled as beads on a string. So up at the very top, we have our plain double helical DNA. The next figure down, you have what's referred to as the chromatin, which is beads on a string. These individual structures that you see that are the spheres with the DNA wrapped around it are referred to as nucleosomes. So one sphere represents a set of proteins referred to as histones, and we're going to look at this in more detail in the next several slides. Those histones represent a core of proteins where the DNA is wrapped around twice. So if you think about taking a tennis ball, which would represent your histone core, and a ball of string or a stretch of string, and take that string and wrap the string around the tennis ball twice. That represents an individual nucleosome. You then take the string and wrap it around the next tennis ball twice, and then take the string and wrap it around the next tennis ball twice. Each one of the tennis balls with the DNA wrapped around it twice is a nucleosome, and this already compacts the DNA significantly. The next level of organization is referred to as the 30 nanometer fiber. And the 30 nanometer fiber essentially just tightens up the slack that you see in the structures that we were just discussing previously. So if you could tighten up that slack, you could condense the structure even more. The next level down is referred to as the 300 nanometer fiber. And the 300 nanometer fiber is basically taking the 30 nanometer fiber and wrapping it back and forth and back and forth and back and forth to further condense the genetic material. The last thing that I want to mention on this particular slide is the figure down at the bottom. The figure down at the bottom represents the classic chromosome structure that we've all seen multiple times in other classes, in social media, wherever you want to say that you've seen chromosome structure. It's the typical representation. That representation, though, is an example of a chromosome that has been replicated in order for preparation of cell division. So just keep that in mind. And there is an extreme amount of condensation that occurs. Hopefully you remember a little bit from your intro cell bio course that it is during mitosis where chromosomes can be visualized under the light microscope because they have condensed so much. And so it's that level of condensation that you're seeing at the very bottom. That's not the everyday level of organization that you would see. Okay, so as I mentioned on the previous slide, that particular slide was a really nice overview to look at the big picture and how the levels of organization continue to grow. And now what we're going to do is we're going to pick that apart in a little more detail. So as I mentioned again on that previous slide, we know that the DNA is found in the classic double helical shape, but we know that the DNA is found in close association with proteins. Those proteins are referred to as histones, and we're going to look at those in a little more detail in just a second. So this particular structure or level of organization is referred to as the nucleosome, and the nucleosome is the fundamental building block to make chromosomes. The DNA is wrapped around the core of proteins twice. So the figure that you see in the top left-hand corner is a great example of what an individual nucleosome would look like. Nucleosomes, the histones, are a number of proteins that are found in the center of this group of proteins. The histones are small proteins that are positively charged, and it is this positive charge that interacts very nicely with the negatively charged DNA. We covered this concept in the last lecture that DNA is deoxyribonucleic acid. It has a net negative charge, and so these proteins, the histones, have a net positive charge, and the interaction between the positively charged histones and the negatively charged DNA interact very nicely to hold the two of those things together. These histone proteins are composed of lots of lysine and arginine amino acids, which again are positively charged. Each histone core is made up of two of each of the following proteins: a protein known as H2A, a protein known as H2B, H3, and H4. So there are two of each of those in the histone core. This is referred to as an octamer because there are 8 proteins in the core. And every histone also has one protein known as H1. In this figure, it is the green protein that is very different or distinct from the other ones that are part of the center of the histone core. In the bottom figure, you can see an electron micrograph that shows this structure. And this was oftentimes described as beads on a string, in that you have nucleosome after nucleosome after nucleosome with the DNA linking one to the other. Now, each one of the histones, as I mentioned in the previous slide, are small and positively charged. That positive charge allows for the interaction with the negatively charged DNA. In addition, each one of the histones has a portion that's referred to as a tail. And in this particular figure, what you're seeing is the main structure of the histones, sort of as that pie wedge. And you can see them listed along the right-hand side, H2A, H2B, H3, and H4. So again, that little pie piece is the main core of each one of those individual proteins. However, each of the proteins also has a stretch that's not part of the main core. That is the part coming away from them, referred to again as a tail. It is this tail portion of each one of the histones that is the site for extensive modifications that alter the function of each nucleosome, each protein, and each nucleosome. Now, when we talk about modifications, what we're talking about are additions or things that can be removed. Basically, we're talking about components that can be added on or taken off, meaning that it is reversible. These are typically functional groups that can be added to specific locations to have different functions. Now, down in the bottom left-hand corner, you can see the legend that indicates the different types of modifications that can occur to each of the proteins and the specific location, that is, which amino acid is that modification actually occurring at. What we will talk about are methylation, acetylation, phosphorylation, and we will very briefly talk about ubiquitination. All right, so the main three are acetylation, methylation, and phosphorylation. So the first category I want to talk about is probably the most common type of modification that occurs, and that is the addition of the acetyl group. The acetyl group is CH3CO, and this typically occurs to lysine amino acids in the tail. There are a specific group of enzymes or family of enzymes known as histone acetyl transferases, or abbreviated HATs, and there are a family of enzymes that remove these acetyl groups known as HDACs, histone deacetylases. So we have a family of enzymes that can put this acetyl group on, and we have a family of enzymes that can remove the acetyl group. And again, this is the key to modifications, is that you can add them on and take them off. And by adding on or taking off, that modulates or regulates the activity. So again, focusing on acetylation, this usually occurs in regions that are going to become active in transcription. Basically, what it does is open up the chromatin structure. By adding acetyl groups, it neutralizes the positive charge. So let's take one step back. We said that the histones have a net positive charge, and the amino acids have a net negative charge. And it is that negative positive interaction that holds the proteins and the DNA together. If we add acetyl groups to some of those positive charges, we remove the positive charge or neutralize it. And in a sense, we lose that positive-negative interaction that's occurring between the protein and the DNA. So you get a very loose association of the histones with the DNA with acetylation, and that's sort of just a rule of thumb. And in the figure, you can see the little pink boxes indicating where acetylation can occur on individual histones and the specific amino acid that it can occur at. The next type of modification I want to talk about is methylation, and that's the one indicated by the little blue arrow or blue triangle. Methylation involves the addition of methyl groups, which is CH3. Typically, methylation also neutralizes the charge on lysines and arginines, but depending upon where and specifically which location the methyl group is added will have differing effects. So there's not a one common rule like what we just talked about with acetylation, where acetylation in general allows for neutralization of charge and allows for loosening up that association of the DNA and the protein. Methylation, it depends again where the methyl group is added, what effect it's going to have. So no one blanket rule here for methylation. Some examples, methylation allows for repression of transcription, whereas in some cases methylation will loosen up that association to allow more easy transcription to take place. Phosphorylation is the next modification that I want to talk about, and that's indicated with the yellow circle on this particular figure. This is the addition of a phosphate group, and the phosphate group addition occurs to serine and threonine amino acids. And you can see on the figure there's very few locations that phosphorylation will actually occur. But what phosphorylation does is typically make the chromosomes much more compact as they get ready or prepare for mitosis and meiosis. So a few slides back, that overview slide at the very bottom of that figure, I mentioned that classic chromosome structure that we typically look at or refer to as chromosome structure. That is a completely condensed chromosome that we could visualize under the light microscope as the cell is getting ready to divide. During interphase, cells, the chromosomes would not be so condensed. We wouldn't actually be able to see structures with the light microscope. So it's this complete further compaction that phosphorylation causes that allows for that preparation before mitosis or meiosis. And then lastly is ubiquitination, and this is the addition of ubiquitin. This is actually a large group, and what you can see on the figure, there's only 2 locations that are indicated that can have the ubiquitin added to it, and that is to H2A and H2B in a different location than the tail structure that we've been focusing on. Typically what happens with ubiquitination is that it allows for transcription activation. However, the most well understood role of ubiquitination or ubiquitination is to play a role in DNA repair processes. And so we will talk about that in a little more detail when we talk about DNA damage and DNA repair. So moving up from nucleosome structure, the next level of organization is referred to as the 30 nanometer fiber. And it's called the 30 nanometer fiber because it's 30 nanometers in diameter. And in this particular figure, what I want to highlight for you is what's referred to as the zigzag model, which is pretty well accepted for the structure of the 30 nanometer fiber. And the idea here is that if you take your nucleosomes and then begin to fold those nucleosomes back and forth in sort of a zigzag pattern, what you get is the 30 nanometer fiber. So essentially what's happening is that you have your DNA that's in between each one of the nucleosomes sort of pulled tighter so the nucleosomes are butting up one against the other. And hopefully you can see that with each one of these figures that you see in this particular slide. One of the things I want to back up and talk a little bit about is the H1 protein. So the H1 protein is part of the nucleosome. Again, it was the one that I highlighted for you in the greenish color that was distinct from the nucleosome core. H1 is thought to sort of lock the DNA in position. So if you go back and you look at that particular figure, you'll see that protein sort of locking the DNA in position. In addition, the H1 protein has been demonstrated to be essential for forming this 30 nanometer fiber. And in the top figure, you can really clearly see that with the highlighted pink color indicating the H1 protein, and that there's a tight association of neighboring H1 proteins that again draw those nucleosomes closer together, basically butting them up against one another so there's no more slack in the DNA in between nucleosome structure. So this H1 protein is essential for this 30 nanometer fiber. We also know that the histone tail modifications play a role in allowing for this level of condensation to take place as well. The next level of organization is referred to as the 300 nanometer fiber. And the 300 nanometer fiber basically takes the 30 nanometer fiber and folds it up back and forth, and again further condenses it. In the figure that you see on the left, which is the cartoon version, basically it sort of looks like a bunch of petals along a stretch or a strand. That strand is referred to as the nuclear scaffold, and the nuclear scaffold is the attachment point for these loops of 30 nanometer fiber. On the right-hand side, you can see how complex it is, where there is this back and forth petal-like structure with the attachment point at the nuclear scaffold in the center. So the last two slides focuses on some additional terms and structural features of the genome. This is referred to as heterochromatin versus euchromatin, and these are clearly distinct structural differences in the genomic material and the organization of the genomic material in the nucleus of the cell. So the two figures that we're going to look at, this slide and the next slide, really just highlight that so that you can see it in an electron micrograph image overall. All right, so heterochromatin is actually much more densely packed, and euchromatin is much less densely packed. And you can see that in the figure overall, where it's labeled heterochromatin, it's much darker, which indicates it's more densely packed, and euchromatin is sort of lighter, which indicates that it's less densely packed. So heterochromatin is found in parts of chromosomes where there are few or no genes. Oftentimes it's regions of centromeres and telomeres, which are structural characteristic components of the chromosome that again are very densely packed. These regions tend to be enriched with intergenic sequences, a concept we talked about in the last lecture. And interestingly, genes that may be present in these regions are oftentimes inactive, meaning they're not being transcribed. And again, that goes to this concept of having it much more tightly packed. It's very hard for the enzymes involved in transcription to get in at the DNA to allow transcription to occur. In contrast to that, euchromatin is associated with parts of chromosomes that contain many genes. It's much more loosely packed, and it's found in that 30 nanometer fiber level of organization. So heterochromatin is more of the 300 nanometer fiber level of organization. Euchromatin is more of the 30 nanometer fiber level of organization. The loops are often found near nuclear pores, and this is because this portion of the genome tends to be very transcriptionally active. There is an increase in the amount of acetylation that you find in these regions, and again, that allows for loosening up the association to allow those enzymes to get at the DNA to allow for transcription to occur. And this is just a second figure that highlights both heterochromatin and euchromatin for you a little more distinctly. It's just a different image. And again, you can really clearly see those darker regions that are the heterochromatin, more tightly packed, versus the lighter regions, which is the less densely packed euchromatin overall.